Now we will conclude Lecture 5 with Presentation 3. So, primates can be classified by traits associated with a unique feeding adaptation, habitual food-to-face feeding. There is another trait associated with being primate. 99% of all primate species are fruit freaks. Why? What peculiarity is shared by guinea pigs, capybaras, and most primates? These mammals have no or only partial ability to synthesize their own vitamin C within their bodies. Most mammals make vitamin C in their livers. Thanks to a genetic mutation, all haplorhines, tarsiers, monkeys, apes, and humans, lack the enzyme necessary for vitamin C synthesis. We get so far in the process and then we get to the final stages and we can't complete the job because we don't have that enzyme. Strepsirrhines, lemurs, lorises, and galagos appear to produce inadequate amounts of vitamin C. Interestingly, a separate mutation resulted in the same problem, but involving a different gene, in capybaras and guinea pigs. And thank heavens we now understand that this was a separate genetic mutation, because for a while there we wondered if we were closer to the rodents than we had previously thought. Vitamin C deficiency results in the dietary disease known as scurvy. Scurvy symptoms. Vitamin C is required for collagen production and iron absorption. Vitamin C deficiency results in lethargy and joint pain, so it feels like you've got the flu, anemia, bleeding gums, dental loss, skin hemorrhages, corkscrew hairs, bleeding nail beds, malformed nails, and an inability to heal. For an individual with scurvy, even a paper cut can leave the skin open to infection. All of this adds up to infection vulnerability and impaired digital use. Vitamin C deficiency kills primates. And think about this, anything that impacts use of the digits will impact the ability to get fruit in the wild. So, dietary sources of vitamin C, fruit is an outstanding source. The liver of mammals is excellent. Some insects have vitamin C, and most reptiles have vitamin C in their kidneys. Now, for me, given those choices, I'm going to go for fruit every time. So, all primates are characterized by diets high in vitamin C. The tarsier is the only primate that has never been observed consuming fruit in the wild. However, it is entirely faunivorous and probably obtains vitamin C from animal matter, mammal livers, reptile kidneys, and insects. And remember, it doesn't have to eat a lot of those foods because it's a very small-bodied animal. Therefore, all primates utilize their specialized primate feeding traits to maintain vitamin C heavy diets. The primary source of vitamin C for most primates is fruit. There is a strong mutualistic ecological interaction between non-human primates and fruit. Mutualism involves species interaction in which both of the interacting species profit from the interaction. Fruit is not easy to obtain. It is a patchily distributed and clumped resource, variably mature, and competition for fruit in the rainforest is quite high. Fruit is the primary source of primate dietary vitamin C and prevents scurvy-related death. Young primates learn how to obtain fruit while foraging with their mothers. Interestingly, wild fruit needs primates. Primates are major seed dispersal agents. In addition, some fruit cannot germinate unless it has been through the intestines of a large-sized mammal. We have employed discrete diagnostic traits to explain why primates are included in the class Mammalia and in the order Primates. Discrete taxonomic traits result in discrete taxonomic units. We have seen that inclusion in the order Primates requires possession of the majority of its taxonomic traits. For example, humans have 5 of the 6 adaptive primate traits. We don't have a divergent hallux, and therefore we are primates. We've got the rest of those traits, plus we have a non-adaptive primate trait, a petrosal auditory bulla, which I will discuss later on. And we understand that convergence can result in the possession of one primate adaptive trait, but this does not support placement of cats and cows in the order Primates. We are almost ready to complete our classification task. Our focus is biased again toward human taxonomic units. Sister taxa or sibling lineages are outgroups related at higher taxonomic levels but distinct at lower levels. Comparison between sister taxa is a useful tool for learning classification. So remember that we will have one set of traits and the outgroup will not have those traits. Many people hold the misconception that taxonomic classification is set in stone. Nothing could be further from the truth. 1. All classification is based on a series of hypotheses about the relationship between taxa. 2. Classification can and absolutely must be reconsidered when new data fail to support hypotheses. And 3. Since new data are constantly discovered, this means that classification is in a continuous state of reassessment. For example, the traditional and outdated primate classification had 2 suborders based largely on behavioral data. Take a look at these. 1. There was the suborder Prosimii with lorises, galagos, lemurs, and tarsiers. The traits, mostly small-bodied except those that weren't, mostly nocturnal except those that were diurnal, mostly insectivorous except everybody else. And then the second suborder was Anthropoidea, with monkeys, apes, and humans. Traits, mostly large-bodied, except those that weren't, mostly diurnal, except for those that were nocturnal, and mostly not insectivorous, except for all of the ones that had other types of diets. Note the term mostly. These were not discrete traits. New lines of evidence that include fossil data recovered by paleontologists, molecular analysis by geneticists, and the discovery of previously unknown primate species by field primatologists have resulted in new hypotheses regarding primate classification. This has had particular importance for our understanding of tarsiers. Today, primate classification based on new data indicates that, despite behavioral similarity to lemurs and lorises and galagos, tarsiers are most closely related to monkeys, apes, and humans. I just want to speak for a moment to students who are co-enrolled in the primate course ANTH 370. Please note that this course employs a less rigorous classification of the primates with a focus on human taxonomy at the introductory level. Therefore, it differs in aspects of taxonomy and level of detail. So let's start out by taking a look at the suborders within the order Primates. So who are they, where are they, and what? So we have 2 suborders, the Strepsirrhini, which includes the lemurs of Madagascar, the lorises of Africa and Asia, and the galagos or bush babies of Africa. And then we have the Haplorhini suborder, which includes everyone else. We can use diagnostic traits, these are discrete traits, to separate the two groups. I'll go back and forth across the slide and I'll point out some of these features as we move through. So strepsirrhini traits. 1. Strepsirrhines, lemurs, lorises, and galagos have open eye orbits. See how you can see behind the eye here? The haplorhines, in contrast, the tarsiers, monkeys, and apes and humans, have closed eye orbits. They have a true eye socket. Okay, the strepsirrhines, lemurs, lorises, and galagos have wet noses. How can we tell this from looking at a skull? Look right here, right in this area between the upper incisors. You'll see a gap. That's because moist tissue from the nose inserts into the roof of the mouth for the strepsirrhines. This allows them to taste and smell the world at the same time. In contrast, the haplorhine nose is dry. There is no gap here between the central incisors. So all of the moist tissue is kept inside of the nose, and we refer to it as a dry nose. External lacrimal duct in the strepsirrhines. What is that? Well, if you look out here, the lacrimal duct is the duct that produces the lacrimal fluid that you know of as tears. It's outside on the muzzle in a strepsirrhine primate, as it is in most mammals. For the haplorhine primates, though, it's inside the eye orbit, just like it is on you. And this washes over the eyes and keeps them moist. Remember, this is probably related to our heightened dependence on visual adaptations. And then finally, strepsirrhines, lemurs, lorises, and galagos, get to have tooth combs. And that is right down here. You'll see procumbent incisors and canines sticking out. For the haplorhines, we don't get tooth combs, and our lower teeth are more vertically placed than you see in the strepsirrhines. So, once again, for the strepsirrhine, open eye orbit, a wet nose, an external lacrimal duct, and a tooth comb. For the haplorhines, we have closed eye orbits. We don't have wet noses. We've got dry noses. We have an internal lacrimal duct, and we don't have tooth combs. Now we're ready to get rid of the strepsirrhines. We'll also set aside the tarsiiforms, and we're going to focus into the two infraorders that are sister taxa and of most concern to anybody who wants to know about apes and humans. So, our infraorders, we have Platyrrhini for the New World monkeys of tropical America, and we have Catarrhini for apes and humans, as well as Old World monkeys. So again, let's go back and forth here. The Platyrrhini traits, 2, 1, 3, 3 dental formula. Allow me to explain this. If you take the mouth of a primate below and above, and then you split it in half right down the midline and across here, we have 1/4 of the mouth here, another quarter here, and another quarter here and here. And what a dental formula is asking about is how many incisors and canines and premolars and molars are in each quadrant of the mouth. It's really very simple. All you have to do is count teeth and you can figure this one out. So let's take a look at this here. 2, 1, 3, 3 in a Platyrrhine New World monkey. So we've got 1 and 1, 2 incisors, 1 canine, 1, 1 premolar, 3 premolars, and then we've got the molars back here. So we have 2 incisors, 1 canine, 3 premolars, and 3 molars in a Platyrrhine primate. How about for the catarrhines? Well, we have 2 incisors, 1 canine, 2 premolars, and 3 molars. So our dental formula is 2, 1, 2, 3. We have fewer premolars than the Platyrrhine New World monkeys do. The catarrhines again are the Old World monkeys, apes, and humans. The next feature we'll look at is zygomatic parietal cranial bone contact. Okay, the zygomatic bone is the cheekbone right here, and the parietal bone makes up the wall of the brain case. These two contact one another in the platyrrhine primates, so we say that they have zygomatic parietal contact. If we go over to the other side and we take a look at the catarrhine primates, this is a zygomatic. It does not touch the parietal bone right here. Instead, what happens is our single frontal bone swings around and down, and the bone that comes underneath the skull, it's shaped like a butterfly. So in Latin, that's sphenoid, comes up like this and touches. So our point of contact is right here. We have frontal sphenoid cranial bone contact. And then finally, the platyrrhines, like most mammals, do not have ear tubes. You have the entrance into the hearing area, the outside opening, so it's the external auditory meatus. It's surrounded by a circle of cartilaginous material, and the hearing cup is right behind that entrance. For the catarrhines, in contrast, here is that opening into the ear region, the external auditory meatus, and then we have a long tube that goes in like a tunnel and comes into the hearing cup, which is placed closer to the midline of the skull, beneath the skull. So, one more time, for the Platyrrhine New World monkeys, we have the dental formula 2, 1, 3. 3, trying to get rid of these here. We have zygomatic and parietal contact and no ear tubes. For the catarrhini, the Old World monkeys, apes, and humans, the dental formula is 2, 1, 2, 3. So fewer premolars. And instead of zygomatic parietal contact, what we have is frontal sphenoid cranial bone contact. And instead of having the old style mammalian opening into the ear cup, what we have is an extension with bony ear tubes. So right through here. All right. And now we're ready to go into that catarrhini infraorder and sort out the Cercopithecoidea Old World monkeys from the Hominoidea apes and humans. Let's take a look at this. So here we have quite a bit of information to look at. Starting with the Cercopithecoid Old World monkeys. These are of Africa and Asia, macaques, baboons, and face-masked monkeys, and many more. And what we see is that they have a bilophodont molar. And what the heck is a bilophodont molar? The molar has 2 parts, that's the bi, and on top of this molar, so it's in 2 parts, 1 and 2, we have 4 cusps to look at here. Now, for the hominoids, apes and humans, they have a round ovoid shaped tooth. So notice it's very circular. It has 5 cusps on it. 1, 2, 3, 4, 5. And then right down the seams we have a letter Y. That is a Y5 molar. Now, in the Cercopithecoidea, if we look at the silhouette of the skull, what you will find is that the face is much larger than the brain region. For the hominoids, apes and humans, the brain is larger than the facial region. And finally, all cercopithecoid Old World monkeys are standard issue mammalian quadrupeds, 4 limbs, and they always have a tail. For the hominoids, apes and humans, we don't have quadrupedal adaptations. Instead, we have some bizarre adaptations of the upper body that allow us to engage in suspensory behavior using our hands. And we don't have a tail. So, one more time, for the cercopithecoid Old World monkeys, they have bilophodont molars, that is a 2-parted tooth with 4 cusps, and they have a face that is larger than the brain, and they have quadrupedal adaptations with a tail. For the hominoid apes and humans, what we have is Y5 molars, so here's the Y, and here are the 5 cusps that give it its name. And also notice that that tooth is round to ovoid in shape. The brain is larger than the face, and we have suspensory upper body adaptations, and all of that comes together with not having a tail. So that leaves us with one question. We've sorted out how we fit into the order Primates, how our suborder Haplorhini is different from the suborder Strepsirhini. We've gone in and focused on 2 infraorders, the Platyrhini for the New World monkeys and the Catarrhine that we belong to for cercopithecoid Old World monkeys and hominoids. And then we went down to the superfamily level and we separated the hominoidea from the cercopithecoidea. Well, now, how do we separate ourselves from apes? Hominoid classification is relatively easy to understand. First of all, let's go to the bottom of the page. Notice that in Asia there are 2 apes, the gibbon and the orangutan, and in Africa we have the gorilla and the chimpanzee. Not surprisingly, since humans have an African origin, our closest relatives are the African apes, and of these we are closest to the chimpanzee. Now, our superfamily that holds all apes and humans is Hominoidea, and there are 2 families. One family, Hylobatidae, only holds the gibbon ape. The Hominidae holds the 3 great apes, orangs, gorillas, and chimpanzees, and humans. Within the Hominidae, there are 2 subfamilies, Ponginae, which just holds the orangutan, and then Homininae, which holds the gorillas, chimps, and humans. Within the subfamily Homininae, we have 3 tribes, Gorillinini for the gorillas, Panini for the chimpanzees, and finally Hominini for the humans. If we want to ask about who's in which group, we can use the short terms for these names. Hominoids for Hominoidea includes all apes and humans. Hominids for Hominidae holds the great apes and humans. Hominines for Homininae holds the African apes and humans. And finally, hominins only refers to humans. There are 2 major areas that separate apes and humans, and the first of these involves skull features. The second involves the locomotor anatomy. And so let's start out with the skull. The first thing I want you to notice is that chimpanzees, like most primates, have very large canine teeth. These canine teeth extend by long roots into the jaw. Down below, you've got another large canine tooth, and it has a long root as well. Well, because of that, they need some place to place the roots for these large canine teeth, and this reshapes the jaws in certain ways. In humans, we have such small canines that they almost look like incisors, and the roots are equally small. Same thing with the canines down below. So right here. All right. let's get rid of that, and then we'll walk through the features, because the features of the skull relate to these differences in canine size. So for the ape, they have prognathic faces that stick out and away from the skull. For the human, the face is very orthognathic, meaning that it's small and tucked in beneath the skull itself. The chimpanzees have large canines, and remember that comes with a large canine root. So here's another large canine here on the lower jaw. And for humans, we have very small canines that are almost difficult to tell apart from the incisors. Now, for the chimpanzees, with those large canines with large canine roots, their tooth rows are parallel to one another. And for humans, because we don't have to accommodate large canine roots, our jaws are shaped like a parabolic arch, so like the St. Louis arch. All right, now the next trait to pay attention to that separates apes and humans involves information from below the belly button. So up above, we are very much alike in terms of our suspensory, long upper-limbed adaptations. But from the belly button down, we are quite distinct. Only humans have the shapes associated with bipedalism. So we are bipeds, apes are not. So one more time, for the apes, apes have prognathic faces that stick away from the skull. They have large canines with large canine roots, and they have parallel tooth rows, and they do not have bipedal adaptations. Humans, in contrast, have orthognathic faces tucked in beneath the skull, not sticking out, very small canines, and parabolic tooth rows. And while, just like apes, we have those suspensory upper body adaptations from the belly button down, we have bipedal adaptations, and that is one of the key features of being human. So the importance of bipedal adaptations is that among primates, only humans have bipedal adaptations and bipedal traits. This allows us to walk a mile on 2 feet. No other primate can do this. So starting at the top, let's use another color here. The pelvis for a bipedal human is bowl-shaped. It's very short from top to bottom. And the femur has a medial angle, meaning that the thigh bone goes in naturally to the center or midline of the body, so toward the midline. And then the tibia has a horizontal plateau. We don't have side-to-side shimmy or twist in our knee. The foot is very short, and it has multiple arches across the bottom of it, multiple arches. And then finally, the hallux or big toe cannot grasp. It is adducted with a D instead of being abducted, taken away from the other toes with a B. So one more time, human adaptive traits associated with bipedalism include a short bowl-shaped pelvis, a femur with a medial angle that allows us to bring the knee in underneath the midline of the body. And we have a tibia with a horizontal plateau. Our foot is short in length and it has multiple arches. And our big toe cannot grasp. Our hallux is not abducted, it is adducted with a D. Now you are ready for Lecture 6, Primate Adaptations.